Cytoplasm. Cytoplasm, the cellular material outside the nucleus, but inside the plasma membrane, is about half cytosol and half organelles. Cytosol. Cytosol consists of a fluid portion, a cytoskeleton, and cytoplasmic inclusions. The fluid portion of cytosol is a solution with dissolved ions and molecules, and a colloid with suspended molecules, especially proteins. Many of these proteins are enzymes that catalyze the breakdown of molecules for energy or the synthesis of sugars, fatty acids, nucleotides, amino acids, and other molecules. Cytoskeleton. The cytoskeleton supports the cell and holds the nucleus and other organelles in place. It is also responsible for cell movements, such as changes in cell shape and the movement of cell organelles. The cytoskeleton consists of three groups of proteins: microtubules, actin filaments, and intermediate filaments. Microtubules are hollow tubules composed primarily of protein units called tubulin. The microtubules are about 25 nanometers in diameter, with walls about 5 nanometers thick. Microtubules vary in length but are normally several micrometers long. Microtubules play a variety of roles within cells. They help provide support and structure to the cytoplasm of the cell, much like an internal scaffolding. They are involved in the process of cell division and in the transport of intracellular materials, and they form essential components of certain cell organelles, such as centrioles, spindle fibers, cilia, and flagella. Actin filaments, or microfilaments, are small fibrils about 8 nanometers in diameter that form bundles, sheets, or networks in the cytoplasm of cells. These filaments have a spider web-like appearance within the cell. Actin filaments provide structure to the cytoplasm and mechanical support for microvilli. Actin filaments support the plasma membrane and define the shape of the cell. Changes in cell shape involve the breakdown and reconstruction of actin filaments. These changes in shape allow some cells to move about. Muscle cells contain a large number of highly organized actin filaments, which are responsible for the muscle's contractile capabilities. Intermediate filaments are protein fibers about 10 nanometers in diameter. They provide mechanical strength to cells. For example, intermediate filaments support the extensions of nerve cells, which have a very small diameter, but can be a meter in length. Cytoplasmic inclusions. The cytosol also contains cytoplasmic inclusions, which are aggregates of chemicals, either produced by the cell or taken in by the cell. End of page 76, beginning page 77. For example, lipid droplets or glycogen granules store energy-rich molecules. Hemoglobin in red blood cells transport oxygen. Melanin is a pigment that colors the skin, hair, and eyes, and lipochromes are pigments that increase in amount with age. Dust, minerals, and dyes can also accumulate in the cytoplasm. The nucleus and cytoplasmic organelles. Organelles are structures within cells that are specialized for particular functions, such as manufacturing proteins or producing ATP. Organelles can be thought of as individual workstations within the cell, each responsible for performing specific tasks. Most but not all organelles have membranes that are similar to the plasma membrane. The membranes separate the interior of the organelles from the cytoplasm, creating subcellular compartments within their own enzymes that are capable of carrying out unique chemical reactions. The nucleus is the largest organelle of the cell. The remaining organelles are referred to as cytoplasmic organelles. The number and type of cytoplasmic organelles within each cell are related to the specific structure and function of the cell. Cells secreting large amounts of protein contain well-developed organelles that synthesize and secrete proteins, whereas cells actively transporting substances, such as sodium ions, across their plasma membrane contain highly developed organelles that produce ATP. The following sections describe the structure and main functions of the nucleus and major cytoplasmic organelles found in the cell. Nucleus. The nucleus is a large membrane-bound structure, usually located near the center of the cell. It may be spherical, elongated, or lobed, depending on the cell type. All cells of the body have a nucleus at some point in their life cycle. See page 91. Although some cells, such as red blood cells, lose their nuclei as they develop, other cells, such as skeletal muscle cells and certain bone cells, called osteoclasts, contain more than one nucleus. The nucleus consists of nucleoplasm, surrounded by a nuclear envelope, composed of two membranes separated by a space. At many points on the surface of the nuclear envelope, the inner and outer membranes fuse to form pore-like structures. And the page 77, beginning page 78, called nuclear pores. Molecules move between the nucleus and the cytoplasm through these nuclear pores. Deoxyribonucleic acid, DNA, is mostly found within the nucleus. Although small amounts of DNA are also found within mitochondria, see page 83, nuclear DNA and associated proteins are organized into discrete structures called chromosomes. The proteins include histones, which are important for the structural organization of DNA, and other proteins that regulate DNA function. During most of the life cycle of a cell, the chromosomes are dispersed throughout the nucleus as delicate filaments referred to as chromatin. During cell division, the dispersed chromatin becomes densely coiled, forming compact chromosomes. At the beginning of cell division, each chromosome consists of two chromatids, which are attached at a single point called the centromere. The kinetochore, a protein structure within the centromere, provides a point of attachment for microtubules during cell division. DNA determines the structural and functional characteristics of the cell by specifying the structure of proteins. Proteins form many structural components of the cell and all the enzymes, which regulate most chemical reactions in the cell. DNA establishes the structure of proteins by specifying the sequence of their amino acids. DNA is a large molecule that does not leave the nucleus, but functions by means of an intermediate ribonucleic acid, RNA, which can leave the nucleus through nuclear pores. DNA determines the structure of messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA, all described in more detail on page 86. The sequence of nucleotides in a DNA molecule that specifies the structure of a protein or RNA molecule is called a gene. Because mRNA synthesis occurs within the nucleus, cells without nuclei accomplish protein synthesis only as long as the mRNA produced before the nucleus degenerates remains functional. The nuclei of developing red blood cells are expelled from the cells before the red blood cells enter the blood, where they survive without a nucleus for about 120 days. In comparison, many cells with nuclei, such as nerve and skeletal muscle cells, survive as long as the person survives. A nucleolus is a somewhat rounded, dense region within the nucleus that lacks a surrounding membrane. Usually, one nucleolus exists per nucleus, but end of page 78, beginning page 79. Several nucleoli may also be seen in the nuclei of rapidly dividing cells. The nucleolus incorporates portions of 10 chromosomes, 5 pairs, called nucleolar organizer regions. These regions contain DNA from which RNA is produced. Within the nucleolus, the subunits of ribosomes are manufactured. See the section, quote, ribosomes, end quote, below. Ribosomes. Ribosomes are the site of protein synthesis. Each ribosome is composed of a large subunit and a smaller one. The ribosomal subunits, which consist of ribosomal RNA, rRNA, and proteins, are produced separately in the nucleolus of the nucleus. The ribosomal subunits then move through the nuclear pores into the cytoplasm, where the ribosomal subunits assemble with mRNA to form a functional ribosome during protein synthesis. Ribosomes can be found free in the cytoplasm or associated with an intracellular membrane complex called the endoplasmic reticulum. Free ribosomes primarily synthesize proteins used in the cell, whereas ribosomes attached to the endoplasmic reticulum usually produce proteins that are secreted from the cell. Endoplasmic reticulum. The outer membrane of the nuclear envelope is continuous with a series of membranes distributed throughout the cytoplasm of the cell, collectively referred to as the endoplasmic reticulum. Network inside the cytoplasm. The endoplasmic reticulum consists of broad, flattened, interconnecting sacs and tubules. The interior spaces of those sacs and tubules are called cisternae and are isolated from the rest of the cytoplasm. The rough endoplasmic reticulum is called, quote, rough, end quote, because it has ribosomes attached to it. The ribosomes of the rough, end of page 79, beginning page 80, endoplasmic reticulum are sites where proteins are produced and modified for secretion and for internal use. The amount and configuration of the endoplasmic reticulum within the cytoplasm depend on the cell type and function. Cells with abundant rough endoplasmic reticulum synthesize large amounts of protein that are secreted for use outside the cell. Smooth endoplasmic reticulum, which is endoplasmic reticulum without attached ribosomes, manufactures lipids such as phospholipids, cholesterol, steroid hormones, and carbohydrates. Many phospholipids produced in the smooth endoplasmic reticulum help form vesicles within the cell and contribute to the plasma membrane. Cells that synthesize large amounts of lipid contain dense accumulations of smooth endoplasmic reticulum. Enzymes required for lipid synthesis are associated with the membranes of the smooth endoplasmic reticulum. Smooth endoplasmic reticulum also participates in the detoxification process by which enzymes act on chemicals and drugs to change their structure and reduce their toxicity. The smooth endoplasmic reticulum of skeletal muscle stores calcium ions that function in muscle contraction. Golgi apparatus, or Golgi apparatus. The Golgi apparatus is composed of flattened membranous sacs containing cisternae or cisternae that are stacked on each other like dinner plates. The Golgi apparatus can be thought of as a packaging and distribution center because it modifies, packages, and distributes proteins and lipids manufactured by the rough and smooth endoplasmic reticulum. Proteins produced at the ribosomes of the rough endoplasmic reticulum enter the endoplasmic reticulum and then are surrounded by a vesicle, or little sac, that forms the membrane of the endoplasmic reticulum. This vesicle, called transport vesicle, moves to the Golgi apparatus, fuses with its membrane, and releases the protein into its cisternae. The Golgi apparatus concentrates and, in some cases, chemically modifies the proteins by synthesizing and attaching carbohydrate molecules to the proteins to form glycoproteins, or by attaching lipids to proteins to form lipoproteins. The proteins are then packaged into vesicles that pinch off from the margins of the Golgi apparatus and are distributed to various locations. Some vesicles carry proteins to the plasma membrane, where the proteins are secreted from the cell by exocytosis. Other vesicles contain proteins that become part of the plasma membrane, and still other vesicles contain enzymes that are used within the cell. The Golgi apparatus is most highly developed in cells that secrete large amounts of protein or glycoproteins, such as cells in the salivary glands and the pancreas. Secretory vesicles. The membrane-bound secretory vesicles that pinch off the Golgi apparatus move to the surface of the cell. Their membranes fuse with the plasma membrane, and the end of page 80, skipping page 81 because it doesn't have text, it just has illustrations and notes. Beginning page 82. Contents of the vesicles, vesicle, are released to the exterior by exocytosis. The membranes of the vesicles are then incorporated into the plasma membrane. Secretory vesicles accumulate in some cells, but their contents frequently are not released to the exterior until the cell receives a signal. For example, secretory vesicles that contain the hormone insulin do not release it until the concentration of glucose in the blood increases and acts as a signal for the secretion of insulin from the cells. Lysosomes. Lysosomes are membrane-bound vesicles that pinch off from the Golgi apparatus. They contain a variety of hydrolytic enzymes that function as intracellular digestion systems. Vesicles taken into the cell fuse with the lysosomes to form one vesicle and to expose the endocytized materials to form hydrolytic enzymes. Various enzymes within lysosomes digest nucleic acids, proteins, polysaccharides, and lipids. Certain white blood cells have large numbers of lysosomes that contain enzymes to digest phagocytized bacteria. Lysosomes also digest the organelles of the cell that are no longer functional in a process called autophagia. Autophagia, self-eating. In other cells, the lysosomes move to the plasma membrane, and the enzymes are secreted by exocytosis. For example, the normal process of bone remodeling involves the breakdown of bone tissue by specialized bone cells. Enzymes responsible for that degradation are released into the extracellular fluid from lysosomes produced by the cells. Diseases of lysosomal enzymes. Some diseases result from non-functional lysosomal enzymes. For example, Pompe disease is a rare genetic disorder that results from the inability of lysosomal enzymes to break down glycogen. The glycogen accumulates in large amounts in the heart, liver, and skeletal muscles, an accumulation that often leads to heart failure. Familial hyperlipoproteinemia. Hyperlipoproteinemia anemia is a group of genetic disorders characterized by the accumulation of large amounts of lipids in phagocytic cells that lack the normal enzymes required to break down the lipid droplets. Symptoms include abdominal pain, enlargement of the spleen and liver, and eruption of yellow nodules in the skin filled with the affected phagocytic cells. Mucopolysaccharides, such as Hurler syndrome, are genetic diseases in which lysosomal enzymes are unable to break down mucopolysaccharides. Glycosaminoglycans So these molecules accumulate in the lysosomes of connective tissue cells and nerve cells. People affected by these diseases suffer mental retardation and skeletal deformities. Peroxisomes are membrane-bound vesicles that are smaller than lysosomes. Peroxisomes contain enzymes that break down fatty acids and amino acids. Hydrogen peroxide, H2O2, which can be toxic to the cell, is a byproduct of that breakdown. Peroxisomes can also contain the enzyme catalase, which breaks down hydrogen peroxide to water and oxygen. Cells that are active in detoxification, such as liver and kidney cells, have many peroxisomes. End of page 82, beginning page 83. Proteasomes. Proteasomes consist of large protein complexes, including several enzymes that break down and recycle proteins within the cell. Proteasomes are not surrounded by membranes. They are tunnel-like structures, similar to channel protein complexes. The inner surfaces of the tunnel have enzymatic regions that break down proteins. Smaller protein subunits close the ends of the tunnel and regulate which proteins are taken into it for digestion. Mitochondria. Mitochondria provides energy for the cell. Consequently, they are often called the cell's power plants. Mitochondria are usually illustrated as small, rod-shaped structures. In living cells, time-lapse photomicrography reveals that mitochondria constantly change shape from spherical to rod-shaped, or even to long thread-like structures. Mitochondria are the major sites of ATP production, which is the major energy source for most energy-requiring chemical reactions within the cell. Each mitochondrion has an inner and outer membrane, separated by an intermembranous space. The outer membrane has a smooth contour, but the inner membrane has numerous enfoldings called cristae or cristae that project like shells into the interior of the mitochondria. A complex series of mitochondrial enzymes form two major enzyme systems that are responsible for oxidative metabolism and most ATP synthesis. See Chapter 25. The enzymes of the citric acid or Krebs cycle are found in the matrix, which is the substance located in the space formed by the inner membrane. The enzymes of the electron transport chain are embedded within the inner membrane. Cells with a greater energy requirement have more mitochondria with more cristae than do cells with lower energy requirements. Within the cytoplasm of a cell, the mitochondria are more numerous in areas in which ATP is used. For example, mitochondria are numerous in cells that perform active transport and are packed near the membrane where active transport occurs. Increases in the number of mitochondria result from the division of pre-existing mitochondria. When muscles enlarge as a result of exercise, the number of mitochondria within the muscle cells increases to provide the additional ATP required for muscle contraction. The information for making some mitochondrial proteins is stored in DNA contained within the mitochondria themselves, and those proteins are synthesized on ribosomes within the mitochondria. The structure of many other mitochondrial proteins is determined by nuclear DNA. However, these proteins are synthesized on ribosomes within the cytoplasm and then transported into the mitochondria. Both the mitochondrial DNA and end of page 83, beginning page 84. Mitochondrial ribosomes are very different from those within the nucleus and cytoplasm of the cell, respectively. Mitochondrial DNA is a closed circle of about 16,500 base pairs, BP, coding for 37 genes, compared with the open strands of nuclear DNA, which is composed of 3 billion BP coding for 30,000 genes. In addition, unlike nuclear DNA, mitochondrial DNA does not have associated histone proteins. Mitochondrial diseases. Mitochondria play a major role in the synthesis of ATP. Each mitochondrion contains a single DNA molecule with at least 37 genes, 13 of which code for proteins that are important for ATP synthesis. The other 24 genes are important for the expression of mitochondrial genes. Therefore, mutations or changes in mitochondrial genes can lead to disruptions in normal ATP synthesis, reducing the amount of ATP produced by the cells. Disorders that result from such mutations are collectively called mitochondrial diseases. The effects of these diseases are most obvious in tissues that require large amounts of ATP, particularly nervous and muscle tissues. As a consequence, the common symptoms of mitochondrial diseases are loss of neurological function and defects in muscular activity. For example, Leber hereditary optic neuropathy results in vision loss due to optic nerve degeneration. Mutations associated with this disorder have been found in the genes that function in ATP synthesis, especially in the cells of the optic nerve. Because of their high energy demands, the cells of the optic nerve are damaged or die because of the lack of ATP. In humans, mitochondria are passed only from the mother to her offspring, because the mitochondria of sperm cells do not enter the oocyte during fertilization. See Chapter 29. Therefore, mitochondrial diseases involving the mitochondrial genes show a pattern of maternal inheritance, in which a mother afflicted with a mitochondrial disease passes it to all of her offspring, but a father suffering from the same disorder passes it to none of his offspring. Centrioles and spindle fibers. The centrosome is, a specialized zone of cytoplasm close to the nucleus, is the center of microtubule formation. It contains two centrioles. Each centriole is a small cylindrical organelle about 0.3 to 0.5 micrometers in length and 0.15 micrometers in diameter, and the two centrioles are normally oriented perpendicular to each other within the centrosome. The wall of the centriole is composed of 9 evenly spaced, longitudinally oriented parallel units or triplets. Each unit consists of 3 parallel microtubules joined together. Microtubules appear to influence the distribution of actin and intermediate filaments through its control of microtubule formation. The centrosome is closely involved in determining cell shape and movement. The microtubules extending from the centrosomes are very dynamic, constantly growing and shrinking. Before cell division, the two centrioles double in number. The centrosome divides into 2 and 1 centrosome, containing 2. End of page 84, beginning page 85. Centrioles moves to each end of the cell. Microtubules, called spindle fibers, extend out in all directions from the centrosome. These microtubules grow and shrink even more rapidly than those of non-dividing cells. If the extended end of a spindle fiber comes in contact with a kinetochore, a specialized region in the centromere of each chromosome, the spindle fiber attaches to the kinetochore and stops growing or shrinking. Eventually, spindle fibers from each centrosome bind to the kinetochores of all the chromosomes. During cell division, the microtubules facilitate the movement of chromosomes toward the two centrosomes. See, quote, cell division, end quote, page 92. Cilia and flagella. Cilia are structures that project from the surface of cells and are capable of movement. They vary in number from 1 to thousands per cell. Cilia are cylindrical in shape, about 10 micrometers in length and 0.2 micrometers in diameter, and the shaft of each cilium is enclosed by the plasma membrane. Two centrally located microtubules and 9 peripheral pairs of fused microtubules, the so-called 9+2 arrangement, extend from the base to the tip of each cilium. Movement of the microtubules past each other, a process that requires energy from ATP, is responsible for movement of the cilia. Di-9 arms, proteins connecting adjacent, end of page 85, beginning page 86. Pairs of microtubules push the microtubules past each other. A basal body, a modified centriole, is located in the cytoplasm at the base of the cilium. Cilia are numerous on surface cells that line the respiratory tract and the female reproductive tract. In these regions, cilia move in a coordinated fashion, with a power stroke in one direction and a recovery stroke in the opposite direction. Their motion moves materials over the surface of the cells. For example, cilia in the trachea move mucus embedded with dust particles upward and away from the lungs. This action helps keep the lungs clear of debris. Flagella have a structure similar to cilia, but are longer, 45 micrometers. Sperm cells are the only human cells to possess flagella, and usually only one flagellum exists per cell. Furthermore, where cilia move small particles across the cell surface, flagella move the entire cell. For example, each sperm cell is propelled by a single flagellum. In contrast to cilia, which have a power stroke and a recovery stroke, flagella move in a wave-like fashion. Microvilli. Microvilli are cylindrically shaped extensions of the plasma membrane, about 0.5 to 1.0 micrometers in length and 90 nanometers in diameter. Normally, many microvilli are on each cell, and they function to increase the cell surface. A student looking at photographs may confuse microvilli with cilia. Microvilli, however, are only 1/10 to 1/20th the size of cilia. Individual microvilli can usually be seen only with an electron microscope, whereas cilia can be seen with a light microscope. Microvilli do not move, and they are supported with actin filaments, not microtubules. Microvilli are found in the intestine, kidney, and other areas in which absorption is an important function. In certain locations of the body, microvilli are highly modified to function as sensory receptors. For example, elongated microvilli in hair cells of the inner ear respond to sound. Genes and gene expression. Heredity is the genetic transmission of characteristics or traits from parents to their offsprings. Genes are the functional units of heredity. Each gene is a segment of a DNA molecule. DNA molecules, along with their associated proteins, form the chromosomes. The sequence of nucleotides in a DNA molecule is a method of storing information that is based on a triplet code. 3, end of page 86, beginning page 87. Consecutive nucleotides, called triplets, form the words of the triplet code, just as a sequence of letters, quote, "See dog run," end quote, or SEEDOGRUN, can be deciphered to mean, quote, "See dog run," end quote. A sequence of bases, such as, quote, "CAT, GAG, TAG," end quote, has meaning, which is used to construct other DNA molecules. RNA molecules, or proteins. A molecular definition of a gene is all the triplets necessary to make a functional RNA molecule or protein. Most chromosomes contain thousands of genes. There are two major types of genes: structural and regulatory. Structural genes are those DNA sequences that determine specific amino acid sequences in proteins, such as enzymes, hormones, or structural proteins such as collagen. Regulatory genes are segments of DNA involved in controlling which structural genes are expressed in a given tissue by determining the structure of proteins and by regulating which proteins are produced by cells. Genes are responsible for characteristics of cells and therefore the inherited characteristics or traits of the entire organism. The production of proteins from the information stored in DNA is called gene expression. Gene expression involves two steps: transcription and translation, which can be illustrated with an analogy. Suppose a cook wants a cake recipe that is found only in a reference book in the library. Because the book cannot be checked out, the cook makes a copy or transcription of the recipe. Later in the kitchen, the information contained in the copied recipe is used to make the cake. The changing of something from one form to another, from recipe to cake, is called translation. In this analogy, DNA is the reference book that contains many recipes, genes, for making different proteins. DNA, however, is too large a molecule to pass through the nuclear envelope to go to the cytoplasm, the kitchen, where the proteins are synthesized. Just as the reference book stays in the library, DNA remains in the nucleus. Therefore, through transcription, the cell makes a copy of the gene, the recipe, necessary to make a particular protein, the cake. The copy, which is called mRNA, travels from the nucleus to ribosomes, the kitchen, in the cytoplasm, where the information in the copy is used to construct a protein, i.e. translation. Of course, to turn a recipe into a cake, ingredients are needed. The ingredients necessary to synthesize a, end of page 87, beginning page 88, protein are amino acids. Specialized transport molecules called transfer RNA, tRNA, carry the amino acids to the ribosomes. In summary, gene expression involves transcription, making a copy of a small part of the stored information to RNA, and translation, converting that copied information into a protein. The details of transcription and translation are considered next. Transcription. Transcription is the synthesis of mRNA, tRNA, and rRNA based on the nucleotide sequence in DNA. Transcription occurs when a section of a DNA molecule unwinds and its complementary strands separate. One of the DNA strands serves as the template strand for the process of transcription. Nucleotides that form RNA align with the DNA nucleotides in the template strand by complementary base pairing. An adenine aligns with a thymine of DNA. Cytosine aligns with a guanine of DNA, and guanine aligns with a cytosine of DNA. Instead of thymine, uracil of RNA aligns with adenine of DNA. Thus, the sequence of bases that align with the TCGA sequence of DNA is AGCU. This pairing relationship between nucleotides ensures that the information in DNA is transcribed correctly into RNA. RNA polymerase is an enzyme that synthesizes the complementary RNA molecule from DNA. RNA polymerase attaches to a DNA nucleotide sequence called a promoter. RNA polymerase, however, does not attach to the promoter by itself. It must first associate with other proteins called transcription factors in order to interact with the DNA. The attachment of RNA polymerase to the promoter causes a portion of the DNA molecule to unwind, exposing the nucleotide sequence for that region of the template strand. Complementary RNA nucleotides then align with the DNA nucleotides of the template strand. The RNA nucleotides are combined by dehydration reactions, catalyzed by RNA polymerase, to form mRNA. Only a small portion of the RNA molecule unwinds at any time. As complementary nucleotides are added to the mRNA, RNA polymerase moves along the DNA unwinding the next portion, while the previously wound section of DNA strands winds back together. When RNA polymerase encounters a DNA nucleotide sequence called the terminator, it detaches from the DNA, releasing the newly formed mRNA. The region of a DNA molecule between the promoter and terminator is a gene. The structure of a gene is more complex than just the nucleotides that code for a protein. Some regions that are transcribed to form mRNA do not code for parts of a protein. The DNA sequence of a gene contains a protein coding region and flanking untranslated regions. During transcription, the sequence of nucleotides in DNA between the promoter and the terminator serve as a template to make an RNA molecule called pre-mRNA. The untranslated regions are end of page 88, beginning page 89. Transcribed into pre-mRNA, but do not code for parts of a protein. Instead, these regions function into the regulation of translation. The protein coding region of RNA contains sections called exons that code for parts of a protein and sections called introns that do not code for parts of a protein. Before pre-mRNA leaves the nucleus, it undergoes several modifications called post-transcriptional processing, which produces the functional mRNA that is used in translation to produce a protein or part of a protein. A 7-methylguanosine cap is added to one end of mRNA, and a series of adenine nucleotides called a poly-A tail is added to the other end. These modifications to the ends of the mRNA ensure that mRNA travels from the nucleus to the cytoplasm and interacts with ribosomes during translation. The introns are removed from the pre-mRNA mRNA, and the exons are spliced together by enzymes called spliceosomes. The functional mRNA consists only of exons. In a process called alternative splicing, various combinations of exons are incorporated into mRNA. Which exons and how many exons are used to make mRNA can vary between cells of different tissues, resulting in different mRNAs transcribed from the same gene. Alternate splicing allows a single gene to produce more than one specific protein. However, the various proteins usually have similar functions in different tissues. Thalassemia. Hemoglobin is an oxygen-carrying protein molecule composed of 4 polypeptides. Thalassemia is a group of genetic disorders in which one or more of the polypeptides of hemoglobin is produced in decreased amounts as the result of defective post-transcriptional processing. The decreased amount of hemoglobin in the blood causes anemia, which reduces the oxygen-carrying capacity of the blood. Genetic code. The information contained in mRNA, called the genetic code, is carried in sets of 3 nucleotides units called codons. A codon specifies an amino acid during translation. For example, the codon GAU specifies the amino acid aspartic acid, and the codon CGA specifies arginine. Although there are only 20 different amino acids commonly found in proteins, 64 possible codons exist. Therefore, an amino acid can have more than one codon. The codons for arginine include CGA, CGG, CGU, and CGC. Furthermore, some codons act as signals during translation. AUG, which specifies methionine, also acts as a start codon, which signals the beginning of translation. UAA, UGA, and UAG act as stop codons, which signal the end of translation. Unlike the start codon, those codons do not specify amino acids. Therefore, the protein coding region of an mRNA begins at the start codon and ends at the stop codon. Translation. The synthesis of a protein at the ribosome in response to the codons of mRNA is called translation. In addition to mRNA, translation requires ribosomes and tRNA. Ribosomes consist of ribosomal RNA, rRNA, and proteins. like mRNA, tRNA, and rRNA, are produced in the nucleus by transcription. The function of tRNA is to match a specific amino acid to a specific codon of mRNA. To do this, one end of each kind of tRNA combines with a specific amino acid. Another part of the tRNA, called the anticodon, consists of 3 nucleotides and is complementary to a particular codon of mRNA. On the basis of the pairing relationships between nucleotides, the anticodon can combine only with its matched codon. For example, the tRNA that binds to aspartic acid has the anticodon CUA, which contributes with the codon GAU of mRNA. Therefore, the codon of GAU codes for aspartic acid. Ribosomes align the codons of the mRNA with the anticodons of tRNA, and then enzymatically join the amino acids of adjacent tRNA molecules. The mRNA moves through the ribosome one codon at a time. With each move, a new tRNA enters the ribosome and the amino acid is linked to the growing chain, forming a polypeptide. The step-by-step process of gene expression at the ribosome is described in detail in Figure 3.43, which I'm not going to show because of copyright. End of page 89, beginning page 90. I will read the notes, but again, I'm not showing illustrations. To start protein synthesis, a ribosome binds to mRNA. The ribosome also has 2 binding sites for tRNA, one of which is occupied by a tRNA with its amino acid. Note that the first codon to associate with a tRNA is AUG. The start codon, which codes for methionine. The codon of mRNA and the codon of tRNA are aligned and joined. The other tRNA binding site is opened. By occupying the open tRNA binding sites, the next tRNA is properly aligned with mRNA and with the other tRNA. An enzyme within the ribosome catalyzes a synthesis reaction to form a peptide bond between the amino acids. Note that the amino acids are now associated with only one of the tRNAs. The ribosome shifts position by 3 nucleotides. The tRNA without the amino acid is released from the ribosome, and the tRNA with the amino acids take its position. A tRNA binding site is left open by the shift. Additional amino acids can be added by repeating steps 2 through 4. Eventually, a stop codon in the mRNA, such as UAA, ends the process of translation. At this point, the mRNA and polypeptide chain are released from the ribosome. Multiple ribosomes attach to a single mRNA to form a polypeptide. As the ribosomes move down the mRNA, proteins attached to the ribosomes lengthen and eventually detach from the mRNA. And this is the translation of mRNA to produce a protein. That is the end of page 90, beginning page 91. Many proteins are longer when first made than they are in their final functional state. These proteins are called proproteins, and the extra piece of the molecule is cleaved off by enzymes to make the proprotein into a functional protein. Many proteins are enzymes, and the proproteins of those enzymes are called proenzymes. If many proenzymes were made within cells as functional enzymes, they could digest the cell that made them. Instead, they are made as proenzymes and are not converted to active enzymes until they reach a protected region of the body. Such as inside the small intestine, where they are functional. Many proteins have side chains, such as polysaccharides, added to them following translation. Some proteins are composed of 2 or more amino acid chains that are joined after each chain is produced on separate ribosomes. These various modifications to proteins are referred to as post-translational processing. After the initial part of mRNA is used by a ribosome, another ribosome can attach to the mRNA and begin to make a protein. The resulting cluster of ribosomes attached to the mRNA is called a polyribosome. Each ribosome in a polyribosome produces an identical protein, and polyribosomes are an efficient way to use a single mRNA molecule to produce many copies of the same protein. Regulation of gene expression. Most of the cells in the body have the same DNA. The transcription of mRNA in cells is regulated, however, so that all portions of all DNA molecules are not continually transcribed. The proteins associated with DNA in the nucleus play a role in regulating the transcription. As cells differentiate and become specialized by functioning during development, part of the DNA becomes non-functional and is not transcribed, whereas other segments of DNA remain very active. For example, in most cells, the DNA coding for hemoglobin is non-functional, and little, if any, hemoglobin is synthesized. In developing red blood cells, however, the DNA coding for hemoglobin is functional and hemoglobin synthesis occurs rapidly. Gene expression in a single cell is not normally constant, but it occurs more rapidly at some times than others. Regulatory molecules that interact with the nuclear proteins can either increase or decrease the transcription rate of specific DNA segments. For example, triiodothyronine T3, a hormone released by cells of the thyroid gland, enters cells such as skeletal muscle cells, interacts with specific nuclear proteins, and increases specific types of mRNA transcription. Consequently, the production of certain proteins increases. As a result, an increase in the number of mitochondria and an increase in metabolism occur in these cells. Cell life cycle. The cell life cycle includes the changes a cell undergoes from the time it is formed until it divides to produce two new cells. The life cycle of a cell has two stages, an interphase and a cell division stage. Interphase. Interphase is the phase between cell division. 90% or more of the life cycle of a typical cell is spent in interphase. During this time, the cell carries out the metabolic activities necessary for life and performs its specialized functions. For example, secreting digestive enzymes. In addition, the cell prepares to divide. This preparation includes an increase in cell size because many cell, end of page 91, beginning page 92, components double in quantity and a replication of the cell's DNA. The centrioles within the centrosome are also duplicated. Consequently, when the cell divides, each new cell receives the organelles and DNA necessary for continued functioning. Interphase can be divided into three subphases, called G1, S, and G2. During G1, the first gap phase, the cell carries out routine metabolic activities. During the S phase, the synthesis phase, the DNA is replicated. New DNA is synthesized. During the G2 phase, the second gap phase, the cell prepares for cell division. Many cells in the body do not divide for days, months, or even years. These, quote, resting, end quote, cells exit this cell cycle and enter what is called the G0 phase, in which they remain unless stimulated to divide.